Hello everyone, my name is Hemophilia, and in this video, I will show you how gene therapy works. But first, let's determine what is the meaning of gene therapy. So gene therapy is an experimental technique which uses genes to treat or prevent any disease. In the future, this technique may allow our doctor to treat any disorder by inserting a gene into the patient's cells instead of using any drugs or surgeries. So a lot of researchers are testing several approaches to gene therapy, including by replacing a mutated gene that causes disease with a healthy copy of the gene, inactivating or knocking out a mutated gene that is functioning improperly, and lastly, introducing a new gene into the body to help fight a disease. So this technique is remain risky and the doctors are still working on it since this therapy is only tested for those diseases that have no other cure. These genetic diseases may be classified into 4 categories. First is the single genes changes. It's a single locus or gene which is defective and responsible for the disease, and this is 100% heritable. Examples include sickle cell anemia, hypercholesterolemia, and the cystic fibrosis. The second one is the multi-gene disorders. It consists 2 or more genes which are defective and responsible for the disease, and this is less than 100% heritable. Examples include heart disease, cancer, and the diabetes. The third one is the mitochondrial disorder, where it is a mutation in the mitochondrial DNA which causes disease that affect many of our organ system. And lastly, the chromosomes abnormalities. It is sometimes complete chromosomes or the parts of the chromosome which are either missing, duplicated, or modified in some way. Examples include the Down syndrome. Did you know that there are also types of the gene therapy? As we all know that all the cells of the human body contains gene, and those are the target for the gene therapy. However, these cells can be divided into 2 major categories. First is the somatic cell. So somatic cells are non-reproductive. Somatic cell therapy is viewed as more conservative and safer approach since it affects only the targeted cells in the patients, and this is not passed down on the future generations. In other words, the therapeutic effect ends with the individual who received the therapy. And the other one is the germline, which is egg or the sperm cells. So germline cells results in permanent changes which are passed down to the subsequent generation.
Hello, I'm Josh, and for this video I'll show you what are the gene therapy approaches. There are multiple approaches to gene therapy, and this time I'll show you the somatic gene therapy. Somatic gene therapy can be broadly split into 2 categories. First is ex vivo, which means exterior, where cells are modified outside the body and then transplanted back in again. In some gene therapy clinical trials, cells from the patient's blood or bone marrow are removed and grown in the laboratory. The cells are exposed to the virus that is carrying the desired gene. The virus enters the cells and inserts the desired gene into the cell's DNA. The cells grow in the laboratory and are then returned to the patient by injection into a vein. This type of gene therapy is called ex vivo because the cells are treated outside the body. Next is in vivo, which means interior, where genes are changed in cells still in the body. This form of gene therapy is called in vivo because the gene is transferred to the cells inside the patient's body.
Hi, I'm Aaron Reyes de la Torre. Did you know that there are viruses used as vectors? Let's find out. First is the adeno-associated virus or AAV, a small, non-enveloped virus that packages a linear, single-stranded DNA genome. It usually remains within the target tissues as episomal entities, although integration into the whole cell genome can rarely occur. The other one is the adenovirus, or AD. a non-enveloped virus that contains a linear, double-stranded DNA genome. It has one of the largest capacities for delivering transgenes, is episoma, and offers minimal risk of insertional mutagenesis. AD has a high inflammatory potential. And last is the lentivirus, or LV, is an enveloped retrovirus containing a positive-sense single-stranded RNA genome. It integrates its genome into the chromosome of target cells and can provide long-term expression in dividing cells. LV has a low inflammatory potential.
Hello, I am Althea Marica Tindig and I am going to discuss what is the non-viral DNA carriers. Non-viral delivery methods may be safer than viral methods. Some of these non-viral methods include cationic liposomes, are positively charged lipids that can interact with negatively charged DNA. Naked plasmid DNA injection. A gene construct is made composed of a gene of interest with a promoter to direct protein synthesis. Example of gene therapy clinical trials for diseases include a severe combined immunodeficiency, cystic fibrosis, advanced central nervous system malignancy, mesothelioma, ornithine, Transcarbamylase deficiency, Hemophilia, and Sickle cell disease.
Hi, I am Lian. Did you know that there is a death from gene therapy? The first reported case of death from gene therapy was in 1999. Josie Gelsinger, 18 years old and was suffering from ornithine transcarbamylase deficiency that causes a buildup of ammonia, was given the gene for this enzyme in an adenovirus vector. 4 days after the infusion of the recombinant virus into his liver, he died from complications resulting from a clotting disorder and organ failure due to the adenovirus. However, there is also a success in gene therapy. In 2003, the first commercial gene therapy was approved in China. The therapy, marketed under the name Gendicine, treats head and neck squamous cell carcinoma. This is an injectable therapy that contains an adenovirus vector with p53 tumor suppressor gene. What are some ethical issues surrounded by gene therapy? Because gene therapy involves making changes to the body's set of basic instructions, it raises many unique ethical concerns, including: how can good and bad uses of gene therapy be distinguished? Who decides which traits are normal and which constitute a disability or disorder? Will the high cost of gene therapy make it available only to the wealthy? Could the widespread use of gene therapy make society less accepting of people who are different? Should people be allowed to use gene therapy to enhance basic human traits such as height, intelligence, or athletic ability? Current gene therapy research has focused on treating individuals by targeting the therapy to body cells such as bone marrow or blood cells.
Gene therapy is the transfer of a therapeutic or working copy of a gene into specific cells of an individual. The technique may be used to replace a faulty gene, or to introduce a new gene whose function is to cure or to favorably modify the symptoms of a condition. Gene therapy is still an experimental technique, and much research remains to be done before this approach to the treatment of conditions will realize its full potential.